as our speaker today about calcium signaling and T-cell development. Masa obtained his PhD in medicine in 2000 from Osaka University in Japan. Then he did a nice and very productive postdoc with Tomohiro Kurosaki and identified a role for RAS-GRP3 in PLC gamma-2 signaling in B cells, which for those of us who are interested in antigen receptor signaling was a very nice work published in the Journal of Experimental Medicine, and very important. Then he joined Anjana Rao in Boston at the time, 2004, stayed there for 3 years, and was one of the people who identified stem proteins as subunits and regulators of store-operated calcium channels, the crack channel in T cell. There was a landmark paper that was very well received. In 2008, he moved back to Japan, and he is now an associate professor at Kyushu University. And he has continued to study stim roles in conditional knockouts and so on, and found some very interesting stim function in agonist selection of regulatory T-cell subsets in the thymus. And he has taught me some other interesting tidbits about autoimmune disease functions that are starting to be explored and so on. That's it. With this, I give the phone to him and hope for a nice talk.
Okay. Right. So first, I'd like to thank Carsten for giving me this opportunity. It's a great pleasure for me to talk my recent work here. And I'm Masatsugu Oh-hora at Kyushu University. As Carsten said, I was a postdoc in Anjana Rao's lab in the Harvard Medical School, and then started to study the stop-related calcium entry in terms of the downstream TCR signaling. So I'm going to talk about the role of stop-related calcium entry today. So as already know, so this slide summarizes the T-cell development in the thymus. And CD4, CD8 double positive, CD4, CD8 double negative thymocytes differentiate into the CD4, CD8 double positive thymocytes in the thymus. And at double positive stages, weak or no interactions of the TCR or TCR and peptide MHC complex cannot support the thymocyte survival, leading to the death by neglect. And modality affinity interactions induce the development of functional T cells through positive selections. And high affinity interactions of TCR and peptide MHC complex normally trigger the apoptosis to eliminate the self-reactive thymocyte through negative selections. And in thymocytes, TCR signal strength well correlates with the magnitude and the duration of the calcium influx. So in vitro study demonstrated that the strong TCR signals induced by the peptide promoting negative selection sustain the high concentration intercellular calcium and with large calcium influx, whereas the weak TCR signals induced by the positive induce a peptide promoting positive selection, so induce a small calcium influx and gradually increase the intercellular calcium concentration. So based on the knockout mouse studies, the successful T-cell development requires many TCR signal pathways. Among them, the calcium signaling plays a vital role in the T-cell development. So, for example, the calcineurin B1, calcineurin knockout mice using LCK transgenic mice impaired the positive selections at double positive stages. But in the case of using CD4 CRE mice, this phenotype is ameliorated, suggesting that the importance of calcium signaling in T-cell in positive selection is time dependent. And in contrast to the calcineurin knockout mice, NFAT singly or NFAT to NFAT doubly deficient mice shows no or mild phenotype in positive selections. It is still unclear whether the activation of all NFAT proteins is required for the positive selections. But recently, the barbituric-mediated NFAT2 knockout mice showed impaired the transition of double negative 2 to double negative 3 stages. But this NFAT2 activation is IL-7 JAK3 dependent but not calcium dependent. So once cells are activated, so cells intake calcium by a selective calcium channel or non-selective calcium channels. Among them, the T cell uses the channel gated by the depletion of ER calcium store, which is called store-operated calcium chain, store-operated calcium channels, which I'm going to talk about today. And this picture shows the structure of the regulators of store-operated calcium entry, a clock channel ORI1, and an ER calcium sensor STIM1. And the STIM1 has calcium binding ER hand motif in the ER lumen. And the importance of the store-operated calcium entry is highlighted by the identification of the patients who have non-functional mutations of ORI1 gene and the STIM1 genes. And these patients develop the channelopathies, such as immune deficiency or myopathies and ectodermal dysplasia. And interestingly, the STIM deficient patients only develop the autoimmune diseases such as hemolytic anemia or thrombocytopenia. And as I said, T cell uses store-operated calcium entry as a primary mechanisms for sustained increase of intracellular free calcium ion downstream of the TCR. So after the TCR is activated by the antigen and produce IP3 through the activation of TLC gamma 1, this IP3 binds to the IP3 receptors expressed on the ER and induce a depletion of ER calcium stores. And oops, you should show that this line, but it disappeared. And then this one. Oh, it's delayed. So after the star depletions, calcium dissociates from the ER calcium sensor STIM, and the STIM forms the oligomers. And the oligomerized STIM1 binds to the crack channel and activate the crack channels and induce a sustained store-operated calcium channel. And this store-operated calcium, sustained calcium influx activates the NFAT activation or calmodulin kinase activations, and finally induce the cytokine production and T cell differentiation. So to investigate the physiological